Hello everyone, in this video, our group will be presenting on designing ERBB2 primers. There are 6 parts in this video which are listed in the table of contents. First, let's introduce what is a primer. A primer is short fragments of single-stranded nucleic acid that is used for the initiation of DNA synthesis. Our chosen gene of interest is the ERBB2 gene. ERBB2 gene, also known as HER2, is a proto-oncogene located at the long arm of human chromosome 17 and encodes for a protein in humans. ERBB is abbreviated from erythroblastic oncogene B, a gene isolated from avian genome. HER2 is a member of the human epidermal growth factor receptor family. Amplification or overexpression of this oncogene has a role in the development and progression of certain aggressive types of breast cancer.
Criteria used for primer design. Firstly, primer length. Primer length ranges from between 18 to 22 BP and it has an optimal length of 20 BP. Secondly, primer melting temperature, also known as TM. TM is a temperature where 50% of double-stranded DNA is denatured to form single-stranded DNA. Temperature ranges from between 59 to 65 degrees Celsius and has an optimal temperature of 62 degrees Celsius. Thirdly, the presence of secondary structure, which includes hairpin loop and dimers. Hairpin loop is an unpaired loop of messenger RNA which is structured to look like a loop or U-shape. The presence of a hairpin loop will result in the forward and reverse primer to bind to itself. Therefore, primers with hairpin loops should be discarded. Dimers. Dimers is an oligomer consisting of 2 monomers joined by bonds. The presence of dimer will prevent replication from occurring as it causes the forward and reverse primers to bind to each other. Therefore, primers with dimers should be discarded. Fourthly, PCR product size generated by the primers. Optimal PCR product size ranges from between 70 to 200 BP. If it is too large, it will take very long during extension step in PCR. If it is too small, it may be undetected during agarose gel electrophoresis. Here are some of the other criteria used for primer design: GC content, GC clamp, and intron inclusion.
Next, I will talk about the purpose of amplifying the ERBB2 gene. The reason why we chose this gene is because the ERBB2 gene is amplified or overexpressed in around 30% of breast cancer patients. It is also overexpressed in many other types of cancers such as ovarian, stomach, bladder, salivary, and lung carcinomas. Therefore, by being able to amplify this gene, it provides for the possibility of diagnosing not only for the presence of breast cancer in patients, but also for the presence of many other types of cancers. To design our primers, we firstly used the National Center for Biotechnological Information or NCBI website to get the ERBB2 gene sequence. After finding the gene sequence, we used the PrimerBlast website to create and obtain the primer pairs for the ERBB2 gene. By inputting certain specific parameters that we want for the primers, such as optimal primer length and melting temperature, we are able to create and generate a good pair of primers using PrimerBlast. Lastly, we used GeneRunner, a software used to analyze the forward and reverse primers obtained to detect for the presence or absence of secondary structures such as hairpin loops and dimers in a primer pair.
So the next 3 slides show the primer blast results as well as the oligo analysis, and if you reference the results that we got regarding primer length PCR products, melting temperature of primers, and GC content to the optimum that has been mentioned above, it is of note that all the results are within optimum range. So for discussion, GC clamps are present in both forward and reverse primer, and hairpin loops and dimers, though present, are of a significantly lower melting point than the melting point of the primer. So it means that when the primer is being used at PCR temperature of 60 degrees, the resulting bonds in the hairpin loops and the dimers will denature, and therefore this will not be an issue during the PCR replication.
These are our references and thank you for watching.